We're going to move now on to Stealth Biotherapeutics. We've got Reenie McCarthy joining us today.
Great, thank you so much. And I want to echo Ben's thanks to Connexum and to Zan for inviting us here today. My name is Reenie McCarthy. I'm the Chief Executive Officer of Stealth Biotherapeutics. And I'm pleased to continue the conversation about mitochondrial targeted therapeutics to treat diseases of aging. So we are a publicly traded company. We're listed on NASDAQ under the ticker MITO. So just quickly draw your attention to our forward-looking statements and disclaimers. I know there's been a lot of conversation about this over the course of the conference, but our focus is on mitochondrial targeted medicine to treat diseases of mitochondrial dysfunction, which does include diseases of aging. Mitochondria are the powerhouse of our cells. They take the air we breathe and the food we eat and convert it to adenosine triphosphate, which is the cellular unit of energy. Unfortunately, with aging, spontaneous mitochondrial DNA mutations in our mitochondrial DNA can occur due to factors including obesity, oxidative stress from smoking, pollution, and other factors. And essentially those will damage the mitochondria, leading not only to a reduction in ATP, which obviously affects high energy organ systems, but also to an increase in cellular level emissions of reactive oxygen species or oxidative stress, which drive disease pathology across multiple diseases of aging. And so again, normal healthy mitochondria produce the energy that we need for our organ systems to work properly. Dysfunctional mitochondria actually are involved in multiple downstream disease pathologies. So just as a quick example, on the right, you can see a cell undergoing oxidative stress, and really the cell can't sustain that insult and it eventually succumbs and dies. On the left, with our compound Elamipretide, which is our lead therapeutic compound, we can see a protective effect across oxidative stress conditions. And so looking at electron micrographs of mitochondria, and here you're looking at mitochondria in a mouse model of diabetic retinopathy, but we see this same progression across multiple diseases. On your left is a normal mitochondria, and you can see that it's got healthy inner mitochondrial membrane content, which is really important because it's in the inner mitochondrial membrane that ATP is produced through oxidative phosphorylation. In disease, in the middle, you can see a destruction of that inner mitochondrial membrane content, which after 6 weeks of treatment in this particular model, we see normalization of mitochondrial morphology with Elamipretide. So that inner membrane content is important not just in each mitochondria, but across mitochondrial networks. So if you think of our cells and the mitochondrial networks in them like power grids, the cristae, which is the curves of the inner membrane, should be interconnected across cells. And here you can see that mapped normally in yellow with interconnectivity, and red where there's a disruption in connectivity. And just like in a regular power network, if you start to get a lot of disruptions and disconnections, you start to get energy failure. In our clinical development pipeline, we are focusing across a number of diseases. So for some of our earliest work, we've looked at different orphan diseases, rare diseases where patients have genetic defects affecting their mitochondria, where we can hope to see early signals that we can then eventually translate into larger indications. However, our lead indication in the clinic is in patients with dry age-related macular degeneration, which is the leading cause of blindness in the elderly and is a disease of aging. So what I'd like to do is to take the rest of our time to kind of give you a preview of our different efforts, what we've seen in different organ systems that may be relevant for further development in diseases of aging. So as I mentioned, in dry age-related macular degeneration, we've completed a phase 1 study in 40 patients with this disease. It's a tough disease. It's the leading cause of blindness in the elderly in the Western world. It has a severe impact on quality of life. So it's been described as very comparable to late-stage cancer or end-stage renal disease because it leads to increasing isolation in elderly populations. In our phase 1 study in 40 patients with AMD, we had 20 patients with geographic atrophy, which is an advanced form of dry AMD. And 20 with an earlier form of the disease called drusen. In both cohorts, instead of the expected 2 to 6 letter decline in vision over the 6-month period we treated these patients, we actually saw improvements in vision as well as in reading acuity. And it looks like the drug actually slowed the progression of the disease as well. So we're currently in phase 2B. This is a placebo-controlled trial with 180 patients. It should be fully enrolled by year end, and it's a 1-year trial, so we would expect to have data about a year later. In earlier programs, we've looked at the effect of Elamipretide on other organ systems. So in work done at the University of Washington in old mice, we saw that 1 hour after Elamipretide exposure, we could see an improvement in muscle function and in mitochondrial energy production in the skeletal muscle. So in 40 patients, elderly patients with age-related mitochondrial dysfunction, and this was measured in the thenar muscle group, which is between your thumb and forefinger, which is one of the first muscle groups to decline with aging. So if you start to have trouble opening jars, that could be age-related skeletal muscle mitochondrial dysfunction. In this particular experiment, 2 hours after a single infusion of Elamipretide, we saw about a 30% increase in mitochondrial respiration in the thenar muscle group. And that was associated with an improvement in function. And it's roughly comparable to what was seen in a very similar patient population after about 6 months of endurance and exercise training. Cardiac dysfunction. Mitochondria is a well-known, well-established target for heart failure. So in a study that was done in explanted human heart tissue, this was taken from heart failure transplant patients at the University of Colorado. So it was both ischemic and non-ischemic, as well as congenital heart failure patients that explanted failing human heart tissue exposed to Elamipretide ex vivo. We saw significant improvement in mitochondrial respiration. We've done some studies in heart failure as well. These were small phase 2 studies where we were looking to get early signals to inform later stage development. So there's 2 main forms of heart failure in the United States: heart failure with reduced ejection fraction, where your heart's not squeezing properly. That's typically treated with beta blockers or ACE inhibitors. And there's heart failure with preserved ejection fraction, where your heart isn't relaxing properly, and there's no approved therapies for that. What we saw in a 46-patient trial in heart failure with preserved ejection fraction were trends towards improved cardiac function, particularly during maximal stress, which is when these patients would be most impaired. So we do find that promising for potential further development, and we've seen similar signals in rare disease population called Barth syndrome. Vascular disease is also problematic in the elderly, so both chronic and acute kidney injury. We've seen preclinical signals that are supportive. You're looking at a preclinical model of acute kidney injury where Elamipretide essentially protected against the damage to the microvasculature of the kidney that you can see in the middle, with disease on the right with Elamipretide. It's attenuated, as well as the transient hypoxia. In a study that was done in patients at the Mayo Clinic, we essentially saw the same thing. So improved renal oxygenation and cortical blood flow post-treatment with Elamipretide in a PTRA setting. So neurodegeneration is also another area where mitochondrial dysfunction has been widely implicated. And in preclinical studies, Elamipretide has shown benefit in models of Alzheimer's, Parkinson's, Huntington's, and ALS. Elamipretide does not get into the brain in very high quantities, so we've rationally designed a second-generation molecule called SBT272. We're looking at it in multiple neurodegenerative models. It's also in phase 1 safety testing right now. What we see in 2 different models of ALS, so this is data from a SOD1 model. We've also looked at it in a TDP43 model, is improvement in neuronal protection. And we see the same in a alpha-synuclein model, which is a Parkinsonian type disease. So promising for potential further development in neurodegeneration, we're zeroing in on indications for SBT272. We have a new
2 minutes,
series of
Reenie.
compounds. Okay. Also expected to be first in class, which is 550, also targeting neurodegenerative pathways. So in summary and in conclusion, we do see great promise in mitochondrial targeted therapeutics to treat diseases of aging, potentially even to ameliorate the aging process. Obviously, that would entail further clarification of regulatory pathways. In the meantime, we are hoping that with our dry age-related macular degeneration program, we can bring some benefit to the many elderly patients suffering from this devastating disease of vision loss. And we're actively working on our pipeline to develop new compounds that we can progress into further diseases of aging. Thanks for your time.
Well, Reenie, there's a lot going on there. You guys are busy.
We are.
Yeah, excellent. Well, we've got a question from the floor, which is, are you able to talk to us about the MOA?
Sure, for Elamipretide. Elamipretide diffuses across cell and mitochondrial membranes, and it targets cardiolipin, which is the hallmark phospholipid of the inner mitochondrial membrane. So every time I do a literature search, I see something else that cardiolipin does in the inner membrane, but the primary function is it really establishes the curvature of the cristae, which is where ATP is generated. But it also plays a role in the importation of proteins from the cytosol into the mitochondria, and then as well for getting proteins encoded in the nucleus down into the matrix, because it forms a part of both of those pores. It's also important in signaling mitophagy. So multiple roles for cardiolipin in the inner membrane. It's a phospholipid with several double bonds, so it's very susceptible to peroxidation by oxidative stress. So what you'll see is a bit of a tipping point. If cardiolipin gets damaged, oxidative stress gets greater and greater. So our drug is stabilizing cardiolipin in the inner membrane.
Wonderful. Well, Reenie, that's an excellent presentation. And I'd love to talk more with you about the being a CEO of a publicly quoted longevity business, but we'll have to wrap it up there, I'm afraid. But that's a really great presentation. Thank you.
Great. Thanks for your time.